Okay, so today we're going to start our last module here on the biomolecules of life. So these include carbohydrates, amino acids, proteins, then we'll get into nucleic acids, DNA and RNA. So as you'll see, these sort of smaller functional groups combine really to form biopolymers that make up large biomolecules. Any questions before we get started? So we'll start with carbohydrates. We'll probably take a day or two for these and then move into the amino acids, which we'll spend a little bit longer on. I believe we have 2 exams for this module, so they'll be broken out into different themes based on these different types of functional groups. So starting with carbohydrates, the word is exactly that. When you look at the molecular formula, for every 1 carbon, you have the equivalent of water in it. So it's a hydrated carbon. We typically just call these sugars. And really what they are in terms of functional groups, it's a polyol. So poly means many, OL is hydroxyl group. So it's a carbon backbone with a lot of hydroxyl groups on it. So in photosynthesis in plants, nature takes carbon dioxide plus water and light, and then it makes sugars, and then produces oxygen. So that's why we need a lot of trees on Earth to scrub out CO2 and produce oxygen that we can breathe. So that's what plants do. Humans have to get sugars through our diet. And so that action is called metabolism. And so when you consume a sugar along with the air that you breathe, that basically gets metabolized to CO2. That's what you exhale, plus water. That may be used in other things, plus urination. And then really the energy. So any sort of movement that's happening right now is supplied by metabolism. So that sort of sets the stage for stuff we're not going to talk too much about in this class. If you go into biochem, that's where you'll learn about metabolic cycles. We're going to focus on the structural aspects of carbohydrates. So yet another term that's used is saccharides. And so this, I guess, could be more relevant in terms of like polymer nomenclature, where a carbohydrate is thought to be the repeating unit. And so one sugar is called a monosaccharide. And so if you take that monomer, you link it together, you can get a biological polymer. We'll see some examples of those, what they are when you connect sugars in different ways, what materials do they generate. So the more saccharides you have bonded together, the more they resemble that biopolymer. And so these are kind of the words that are used. So 1 sugar residue is a mono, 2 is a di, 3 is a tri. This range here, 4 to 10, that's called an oligosaccharide. And then once you get past 10, that's a polysaccharide. So oligosaccharides have that sort of shorter chain length to them. And then when you get up into materials like, cotton fiber, for example, is a polysaccharide made from glucose. And so we'll look at other natural sources of these things, like animal shells, chitin, if you've heard that term. So an exoskeleton is made up of polysaccharide. So there's a lot of nomenclature in here, and from the nomenclature you can infer some things about the structure. So we're going to focus on carbon chains that have 3 to 6 carbons as the backbone. And if we think about, shoving some hydroxyl groups on there, then we end up with, these issues of stereochemistry. So we have to consider stereoisomers, their relationships. But based on sort of the chain length, we can use these terms to further understand. So 3 carbons is called a triose. And then if you look at the rest of the columns there in that table, I have these words aldoses and ketoses. And that refers to the functional group of the highest oxidation state on the carbon backbone. So an aldose basically has an aldehyde at the highest oxidation state, and then everything else is at an alcohol oxidation state. A ketose has a ketone. So the carbonyl group has migrated from the terminal carbon of the backbone to the internal carbon. So with 3 carbon atoms, an aldehyde being one of them, the other 2 carbons are going to be alcohols. We can generate 2 aldoses from that. That's because there's one stereogenic center that gives rise to R and S enantiomers. With the same carbon chain length 3, if you have the ketone as the carbonyl, you can basically only draw one ketose. So again, we're going to throw out a lot of different ways of describing these things. Aldose, ketose tells you sort of where the carbonyl is, what functional group it relates to. And then the tri, tetra, penta, hexa, that tells you the number of carbons in the chain. So today we'll look at, starting from the 3 carbon sugars, trioses, we're going to build the chain length up to make tetroses, pentoses, and hexoses. And as you see, for the number of aldoses, it increases quite a bit as we add each carbon to the chain. And again, that's just due to the stereogenic centers that are formed. So important for us to know are these historical designations that are used, big D and big L. So that refers to a very specific carbon on the chain. But keep in mind, for your standard nomenclature rules, you use R and S. So that's the assigning priority. Does it go clockwise or counterclockwise to give you R or S? Additionally, sugars can, so I stress the word here, can have dextrorotatory or levorotatory rotation. So that's the plus or minus sign as measured through a polarimeter. Now, the big D and big L refer to a stereochemical designation of the structure. The plus and minus sign, that's kind of easy to interpret what that means. But there's also another way that's been used to describe these, and that's little d and little l. So plus is little d, that stands for dextrorotatory. That rotates light clockwise. Minus is little l, that's levorotatory. It rotates it counterclockwise. So as you're doing your reading in this textbook or biochem or wherever, you're going to see these nomenclature things pop up quite a bit. So what's another relevance of carbohydrates. Why should we learn them? So in addition to supplying energy through metabolism, carbohydrates are often found on cell surfaces. So on the outside of the cell, there's these carbohydrates that reside. And they are important structural features for recognition. So that's the key word that trigger biological processes to start or stop. When we look at the structure of these carbohydrates, we know that a stereogenic center can be R or S. When you get a lot of them in there, the molecules in one, say, group of diastereomers can have different shapes based on the R and S designations. Those shapes will determine recognition events. So you and I, when we look at something, we perceive this light coming in and our brain does some processing and we recognize. Molecules don't see anything, right? They don't have eyes, so we can't think of it in the same way. But the recognition occurs through the functional groups present in their spatial orientation. So an example of this are the ABO blood groups. So those consist of proteoglycans. So there's another word, glycan, that relates to sugars, in which the protein is embedded in the cell lipid bilayer with the carbohydrate exposed in an extracellular fashion. So the cell lipid bilayer has a polar sort of head group and then a fatty acid side chain. And then that's sort of the external part. Then the internal part is the fatty acid, then the polar side group. So that's the lipid bilayer. Then off the head group are these carbohydrates pointing outside the cell. So there are 4 common blood types, O, A, B, and AB. How many people know their blood type? So you probably know it because you either donate blood or you've had surgery and you have to supply that information. So another reason you have to take it is if you're going to get hitched. The state usually requires blood tests for people getting married. So really it has to do with how things will react in your body. So how are things recognized? Are there going to be adverse events? So what do those letters really mean? So the A and B groups are constructed from the H antigen. Don't know what that is. If you've taken biology, you might know what that is. Go on to take biochemistry, you'll learn what that is. That contain either N-acetyl-galactosamine or D-galactose. So those are 2 sugars. The O group is unmodified. So O is the universal donor, and type AB is the universal acceptor. So if you're undergoing surgery, you either have to go in a couple weeks before and donate your own blood, or they got to make sure that if you do need blood, it's matched for your blood type. So structurally, this is what they look like. So here's the type O, that's unmodified. If we look at it, so this stuff right here, we can see that there's 3 sugar residues. This one to the top left, that's called galactose. This one to the bottom is called fucose. And then this one over here is called N-acetyl. So N means nitrogen, acetyl just means it's an amide, and then it says glucosamine. It ends with that suffix amine because one of the alcohols has been substituted for a nitrogen. And so they all sort of have that, but then let's look at the difference between type A and type B. Can you spot any differences from type O to type A? So if you look at this galactose, this equatorial alcohol, this H has been substituted for another molecule of N-acetylgalactosamine. And then from O to B, what are we doing? That H has just been substituted for galactose. And then the difference between A and B is that secondary position, that N-acetyl part is just OH. So these sugars all have different names. And unless it's the IUPAC name, you really can't infer what the structure is. So like galactose, like if you didn't see the structure, you wouldn't know what to draw. So if you do go on to more biochemistry or biology, it's nice to know these names. And as we'll see, the names of amino acids as well. But I'll show you maybe a trick to help you remember what these things are. We'll see if it takes. So we're going to start from the 3-carbon chain and then review the Fisher projection, because this is the tool that's really going to help us understand the relationships when we start elongating the chain and then looking at the different stereoisomers that can form. So a 3-carbon monosaccharide can be an aldose, that's the aldehyde form, or a ketose, that's the ketone form. Shown in the middle here are the aldose forms. So another way of saying this would be like aldotriose. That tells you it's an aldehyde, it's 3 carbons, and it's a sugar. In the Fisher form that you see, these molecules are acyclic, so it's a linear chain. Carbon number 1 so that's the aldehyde. In the Fisher projection, the highest oxidized carbon is always pointed to the top. That's how it's defined. And then we see there's 1, 2, 3 carbons. So the highest oxidized at the top, carbon 2 is an alcohol, carbon 3 is an alcohol. So we have N equals 3. And if we take N minus 1, that equals 2. So this formula here is for the penultimate carbon. So in general, for any N chain, the N minus 1 is defined as the penultimate carbon. Now when we look at that, it's carbon 2, and the OH group is residing on the right side of the chain. And so that's defined as the big D sugar.
N is the number of carbons, correct?
What?
Is N the number of carbons?
Yes. So the other structure is the enantiomer, and now that penultimate hydroxyl group resides on the left. So that's easy to remember. So L is for left. But again, the D and L, that's for the penultimate carbon only. That's what you're looking at. And so what does the Fisher projection convey to us? So where the chain meets, so like any cross on the horizon, those bonds are defined as being wedges. And then on the vertical, those bonds are really going backwards. So even though like if the chain gets longer and longer, that we're drawing just a line in a plane, that for each successive carbon you look at, the bonds are going backwards and backwards each time. So you can see by the names here, like the IUPAC name. So this D sugar, the stereo center is R. The optical rotation is plus. It's called 2,3-dihydroxypropanol. Or alternatively, we can use that nomenclature, D and then the plus, or this is also called glyceraldehyde. So there's a lot of synonyms for each molecule. And, depending on your curriculum, whether you want to memorize them or not. But you should be able to tell by looking at, this D or L nomenclature. If the rest of the name is the same, then they're enantiomers. So naturally occurring sugars have the D configuration at the penultimate carbon. And unnatural sugars are L. So we basically metabolize D sugars. So your body's not set up to metabolize L sugars in the same regard. And what's interesting about that is, whether you believe in a higher power or evolution, like, why do we only metabolize the D? And so that might become a little bit clearer as we learn about amino acids, peptides, and proteins. The shape of the D sugar is what sort of fits in to the enzymes and proteins that digest them. Yeah.
So how do you determine which one's the penultimate carbon?
So when we're counting this chain, 1, 2, and 3. So if we say n equals 3, we're just subtracting 1. So that gets us to carbon number 2.
I see.
So as we grow out the chain length n, you just subtract 1 and then look at that carbon.
You talk about the carbons going backwards for the R enantiomer. What do you mean by backwards if they are on the
So because that's sp3 hybridized and it's tetrahedral, the horizontal line means that those bonds are coming towards you. And then therefore the ones on the vertical have to be going away from you.
The plane line and the dashed line are the same here?
Well, that's what, when you look at like the Fisher projection itself here in the middle, it looks like from this drawing, carbons 1, 2, and 3 are all in the same plane as the projector, but they're not. Carbons 1 and 3 are actually going back. It's just the way that the learning tool has evolved or developed. That will become important as we grow the chain out, because you don't want to think that because it's shown in a vertical line that they're all, in one plane. So again, there are the enantiomers of the aldotriose. So carbon 2 was the only stereogenic carbon, so that gives rise to the R and the S enantiomers. If the aldose undergoes an isomerization, we learned that the carbon next to a carbonyl is the alpha carbon, right? That can isomerize to a ketose. So this should not say aldo here. That should just say ketose. So now the carbonyl is migrated to an internal carbon. So it's sp2 hybridized. It no longer has a stereocenter. So that's why there's only one of them. And so in these different metabolic pathways, sometimes these isomerizations occur because the ketose is needed and not the aldose. So again, to review, the highest oxidized carbon is always at the top. So that's typically your number 1 carbon. The horizontal lines are hugging you. The vertical lines are running away from you. And then the penultimate carbon, so that's N-1, that determines whether it's D or L. So D to the right, L to the left. So which is the naturally occurring Samuel Jackson? Huh? Samuel L. Jackson is the naturally occurring one, and then his enantiomer is the D. So let's, from those trioses, basically what we're going to do is elongate the chain and get into these different families of sugars. For the most part, what I'm going to teach you is the aldose chemistry. The ketose stuff will conceptually be the same, but I'm not really going to explicitly show that. If we consider a general Newman projection, so if this is carbon 1, the highest oxidized carbon up to N, the last carbon, that has an alcohol. Any internal alcohols, so 2 to N-1, will have what classification? So they all have to be secondary. And the reason I bring that up is, again, this is the highest oxidized carbon. That's a primary alcohol. And then anything in between has to be a secondary alcohol. So the oxidation stuff, we learned quite a bit how you can manipulate oxidation state based on reaction conditions, but then if you have a carbon backbone with a lot of alcohols, how do you differentiate them? The primary will almost always react first, followed by the secondaries. So that's one thing you can think about in terms of a strategy of how to manipulate carbohydrates. So if we think about moving from a triose to a tetrose, how many stereogenic centers does an aldotetrose have? So carbons 2 and 3 will be stereogenic centers. So we have two stereogenic centers. So we say 2 to the n. We found that n was 2. So 2 to the 2 is 4 total stereoisomers. And so we'll go through and look at all their relationships. So these are the D and the L aldotetroses. And conceptually what's happening is we have these pairs. We've talked a lot about stereochemical relationships. If you have one stereocenter, you have R and S enantiomers. When you have two stereocenters, you now have two pairs of enantiomers and then these 4 pairs of diastereomers. Notice the logic here. Again, the aldehyde's at the top of the chain, but let's look at the bottom of the chain. So carbon 4 is your primary alcohol. On the D side, we're keeping that penultimate hydroxyl group to the right. And then what we're changing is the stereochemistry at carbon 2. So it can either go left or right. And then the same thing for the L side. So at carbon 3, the hydroxyl group is to the left, and then the stereocenter is either to the left or the right. So these pairs come from So D-glyceraldehyde can give rise to those D sugars. And then L-glyceraldehyde can give rise to these two. The logic of this is that we know that glyceraldehyde is a 3-carbon sugar. Those 3 carbons are 2, 3, and 4. So we've added a carbon to the top. In glyceraldehyde, this was an aldehyde. So sp2 hybridized carbon, it was a carbonyl. It has 2 faces. You can add to either face. And so that's why you get OH to the left or OH to the right. The relationship between the D sugars is what? Those are diastereomers. And then you see the mirror image relationships. The 2 center sugars and then the 2 outer sugars are enantiomers. Every other relationship is diastereomeric. So as I said, these kind of have weird names to them. So the far left one, that's called D-threose. The inner left one is D-erythrose, and then you have their enantiomers. So one way of kind of reducing the mental burden of this, we use another stereochemical name called epimer. An epimer is a stereochemical relationship where only one CIP designation has changed. So for example, in carbon 2 here, we would call those compounds epimers. So this will make a little bit more sense when we get up to the 5 and the 6 chains, where the epimeric carbon can be anywhere on the chain.
So is the epimers of the like the OH is on the left or the right? If they are changing, then they are called epimers.
Just for one carbon,
Also one,
just for one.
they are very different lifetime. If they have a long period.
Yeah, so basically all of the stereo centers will remain the same, but one will be switched, just one carbon.
Oh.
So the logic here is glyceraldehyde gives birth to threose and erythrose. So that's 4 carbons. Now we're going to go up to 5. So how many stereoisomers total do you think we'll have then? Stereoisomers. So we're going to have 8. So each one of these that you see is going to have 2 kids. And again, the reason for that is we're elongating the chain from the top. So see this aldehyde here? We're going to add a carbon to it. The oxidation state gets reduced from 2 to 1, so it becomes an alcohol. That alcohol can go to the left or the right. So tetrose, each tetrose can have 2 progeny. How many total D and L aldopentoses are there? So there's 8. So that gives us 8 total. So you can sort of see the trend here. Basically, half of them will be D, the other half are L. So of all these possible stereoisomers, again, only the D ones are the naturally occurring ones. So the top 2 structures, there's your erythrose and threose. Notice here that I'm only showing the D sugars. If I put the L ones on there, the structures would be too small. So basically, take these 4, draw their enantiomers, and those are the L sugars. So threose, again, now the penultimate carbon is carbon 3. So again, that groups to the right, so that's the D sugar. So this carbon 1 becomes carbon 2 in the pentose. So notice now it can go to the right or to the left. So erythrose is right, right, and now ribose is right, right, right. The epimer of ribose is arabinose, so it's left, right, right. Yeah.
Do we have to memorize this for the final?
I don't think so. As I said, there's one thing you should know. I'll explain what that is. when we get to the hexoses. But for these, I don't think you have to memorize the structures. So erythros gives rise to ribose and arabinose. Threose gives rise to xylose and lixose. Does any one of these sugars listed up here seem vaguely familiar to you? So ribose, why is that?
It's an RNA.
So it's in specifically, so ribose is an RNA, so ribonucleic acid. DNA stands for deoxyribose, so you've removed one of those OH groups. And so when we get into that part with nucleic acids, you have basically a heterocyclic base that's bonded to this sugar, and then those things form polymers, which you can get RNA strands or the DNA double helix strands. So we're going to continue this logic on, now we have each of these is going to give rise to 2, then plus the L enantiomers. So that's going to give us a total of how many? 16. So 8D, 8L. So we can get 16 hexoses. And this is the parent tree here. Again, these are only the D ones. So ribose, again, we're going to add to this carbon 1, so it becomes carbon 2 in the hexose. We can put it to the right or the left. If it goes to the right, that's called allose. Goes to the left, that's called altrose. Arabinose gives rise to glucose and mannose. Xylose gives gulose and idose. Lixose, galactose, and talose. So which one of these are you familiar with? So glucose. So this is the one I would encourage you to sort of memorize in the sense. So I remember it as in the Fisher projection, right, left, right, right. So getting back to that epimer question, when we look at mannose, so remember the enantiomer of this is the L, so we're not showing that. When we look at mannose, we would say mannose is the C2 epimer of glucose. And so basically what we're saying is now it's left, left, right, right. So again, the epimer only refers to one specific carbon. Everything else has to be the same. So here's the tree shown on one slide, how you can get from the tetrose to the pentose to the hexoses. And then keep in mind, you could draw a mirror plane over there and then have the L series. So let's do some thought experiments based on reaction we know of oxidized carbons. So it says when treated with sodium borohydride, so NaBH4 in methanol, which aldohexose will form a meso compound? So sodium borohydride reacts at what? What functional group? So it has hydride in the name, so it's a reducing agent. So this one's specific for oxidation state 2. So we're dealing with an aldehyde. That has to get reduced to a primary alcohol. So it says when you do that, which one gives you a meso compound? What? Allose. So A, allose is the correct answer. So that aldehyde's been reduced to the primary alcohol. Then within that molecule, there's an internal mirror plane. And then in green, those are your stereocenters. So again, the definition of a meso compound, you have to have stereogenic carbons, and then there has to be an internal mirror plane. As 4 of the 8 so this is the other 4. So same reaction, sodium borohydride and methanol. Which one forms a meso compound? Galactose. So your mirror plane hits between the carbons 3 and 4 sigma bond. So you can see by this sign, it says D plus galactose. The plus means it's rotating the light clockwise in the polarimeter. If you run this reaction and stick in this alcohol, this polyol, in the polarimeter, what's its specific rotation going to be? It'll be 0. So a meso compound, again, even though it has stereocenters, the internal mirror plane will render the specific rotation 0. So here's all 8. So you're going to take all of them, reduce them. So the aldehyde becomes the primary alcohol. 2 of them will converge upon the same compound. Which pair? What'd you say?
Is it B?
B as in boy. That appears to be correct. So altros and telos. So that's the alcohol you get from altros. And then that's the alcohol you get from telos. So how can we say they're the same thing?
The top and bottom rotates freely.
So you're doing a rotation and then they map right onto one another. So this is really part of the chemical logic that Fisher, who developed the Fisher projection, the reason why he developed this was ultimately to prove what the structure of glucose is. So from the start of time until Fisher, people knew glucose existed, but they didn't know the chemical structure, like the exact structure. And so he was the person to prove that, using this tool to help figure out the relationships between these enantiomers, diastereomers, epimers, that sort of thing. And again, what's nice about the Fisher projection, it allows you to figure out these relationships, but also if things are like meso, because if we use the zigzag notation with wedges and dashes, seeing that can be a little bit harder. But Fisher did a lot of these reactions, not necessarily with these conditions, along with the use of polarimetry to help prove what the structure of glucose ultimately is. So same sort of reduction conditions, but now a different element of symmetry. So what two sugars, when reduced, have 180-degree rotational axis of symmetry? So this is not the same question as before. They're not converging onto the same thing. It's saying that within the molecule, if you put an axis 90 degrees to the carbon backbone and rotate it 1/2 turn, everything maps onto itself. So D, mannose, and idose. So let's reduce mannose. And so I'm going to draw a blue dot. One, two. So right there between carbons 3 and 4. So imagine you put an axis where that blue dot is and you rotate it 180 degrees. These alcohols that are on the right become these alcohols on the left. These ones on the left become those. So these last sort of three questions about the meso, the same thing, and then the rotational axis deal with symmetry, so symmetry elements. Any questions on that? Yeah.
I'm a little confused how, if you rotate that, like which way does that rotate?
So for example, if we're rotating like counterclockwise, this OH group would map on to that OH group. And then this one would map onto that one.
You're rotating it anti-clockwise or clockwise?
You can do it either way. So these are the symmetry observations. Allose and galactose form a meso compound, so they have an internal mirror plane. Altros and talose converge upon the same compound, they're identical. Mannose and idose form compounds with, use this term here, C2 symmetry. That just means 180-degree rotational axis. And again, the measurement of specific rotation of all of those sort of products was crucial in helping Fisher figure out ultimately that was the structure of glucose. If we have time over the next lecture or two, I'll talk more about that, but that's sort of an introduction to that logic. So what we're going to do now is look at the other forms of how these carbohydrates are often presented in textbooks. So the straight chain, the Fisher projection is really not present a whole lot under experimental conditions. So these sugars often exist as rings, and they cyclize in a very specific way. And once they do that, it leads to these other relationships that we have to know about. So here's our Fisher projection. At this point, we've talked about the vertical or up and down carbon backbone. Where the horizontal line crosses, that's typically a stereocenter. Those groups are coming out towards you. And then this is what I meant about this kind of misinterpreting what this line means. Just based on this line, it seems like carbon 1 is very far away from carbon 6. But each of these bonds is projected back. So if you imagine, you're looking at this 90 degrees, but if you can take your eyeball sort of over this way and look at it from a different perspective, this is what you would see in the center. That carbon backbone actually sneaks back around on itself. So what we're doing is just changing our perspective of the Fisher projection. There's carbon 1. There's the backbone, and there's carbon 6. So this carbon here actually sits right behind carbon 1. So there's a pretty good proximity. And this is going to lead us into how these things cyclize and then what the name of those sort of drawings is. The first thing we have to do is take carbons 4 and 5 and do a rotation. So I'm going to rotate that hydroxyl group that's down and then sort of stick it in the plane with the aldehyde. So I'm going to rotate that way. So that OH group gets up into the plane. The CH2, that's carbon 6, goes up. I don't know why I drew that H red. We saw in prior module, aldehydes at oxidation state 2 can undergo nucleophilic attack, right? And alcohol is a weak nucleophile. So that can attack from the top or the bottom of that aldehyde face. And then when it does, you end up with 1, 2, 3, 4, 5, a 6-membered ring. That's called the Hayworth projection. So the Hayworth is the ring. And so I think I had it written here. So from our glucose in the center, depending on which face that alcohol attacks, the resulting OH group can go down. So that's called the alpha, or it can go up. That's called the beta. So these things are epimeric. The ring structures are epimeric at carbon 1. But because carbon 1 is a different oxidation state, 2, they're called anomers. Anomers are epimeric acetals. So aldehyde, acetal, acetal, and remember that definition was oxidation state 2, but the carbon's sp3 hybridized. So everything else is the same between the alpha and beta. It's just that carbon 1, whether that OH group points up or down. And you know, we're looking at D-glucose. That's the sugar we use for energy, we metabolize. When you start making polymers of that alpha or beta configuration changes the properties of the polymer quite a bit. And then one more manipulation so that we can get the things that we're used to seeing. The Hayworth projection it's a nice tool that immediately converts the fissure into a ring. But we know already that 6-membered rings form chairs. And so you can take the Hayworth into the chair, and it gives you a more realistic depiction of, are those substituents axial or equatorial? And that's what's important. That's really what determines the recognition event on cell surfaces. And so the way we do that, we take carbon 1, we push it down. Carbon 4, we push up. And then you'll see it's given that nomenclature. We know that carbon 4 is up, carbon 1 is down in that chair form. So what do you notice about, we'll start with the beta anomer. all the substituents on the beta anomer of glucose. So they're all equatorial. And from organic 1, we learned that equatorial substituents are more stable on a ring than axial. So that could be a reason why glucose has evolved to be the chosen sugar for us to metabolize. It's the most stable one. Again, the alpha, beta, those things can isomerize. So that alpha carbon is not locked into an axial OH group. So remember in the Fisher projection, we said glucose was right, left, right, right. In the chair, everything's equatorial. So that's the easy stuff to remember. Then I think, if I'm remembering correctly, I think the C4 epimer of glucose was galactose. So you flip that OH from axial or from equatorial to axial, that draws you galactose. So there's little shortcuts that you can use like that to help you remember the names. So how do we know that these different forms exist? Again, it gets back to the polarimetry experiment. So if you take glucose, dissolve it in water, it's going to set up an equilibrium where these three structures are present. At equilibrium, you only have a trace amount of the open chain or acyclic form. You have the majority of it being the beta anomer, so 64%, and then 36% is the alpha. So at equilibrium, that mixture achieves a rotation of plus 52.7. If you took the pure alpha form, dissolved it and measured it right away, it would be plus 112. But over time, that plus 112 would converge to that number because you're getting these other two forms of it through that isomerization. One other thing I want to mention now that I see it on here. So this says glucopyranose. So the gluco, that comes from glucose, that tells you the sugar. The pyranose, that comes from this structure here. That's called pyran. So glucopyranose is telling you it's glucose in the six-membered ring. Let's see if we remember something here. So we want to form the alpha anomer, so this one over here. And the question is asking you, this alcohol at C5, which face is it attacking to form the alpha anomer? So when we look at the sp2 hybridized carbon of the aldehyde, we can assign priority to the three substituents. I'm gonna squeak this hydrogen in here. And in blue, I'll write the priorities. So oxygen's 1, carbon 2, hydrogen 3. So stated a different way, to form the alpha anomer, do we have to attack the top or the bottom face?
Top.
I don't think so. It's got to be the bottom face. So let's just assign the priority how we're looking at it now. So 1 to 2 to 3, that's clockwise, so that the top face is Re, the bottom face is the psi face. So if that's difficult, it's a matter of kind of your perspective and how you can rotate to see that. But you need this, to make the alpha, you need that OH to go down. So if you rotate kind of about this bond and put this oxygen down, that becomes that oxygen. So in the way it's drawn, this OH has to kind of come from the bottom here. That will plop that H up. So let's see what we got. I think we'll go ahead and stop here because the next run of slides introduces reactions that deal with oxidizing and reducing the chain, and then a couple more symmetry questions.